Hey miss, or anybody else who might have found this video and decided to click on it. Today I would like to talk to you guys about the cell life cycle. There are two main stages in the cell life cycle: the interphase and the M phase. The interphase is basically the cell doing its everyday life functions while preparing for the M phase or mitosis. And the M phase, more commonly known as mitosis, is a division phase. It's all the processes and things that happen that lead the cell to splitting into two identical daughter cells. We will start with the interphase, as it is the phase in which the cell begins its life. The interphase is made up of three separate stages: the G1 or gap 1, S or synthesis, and G2 or gap 2, after which it enters the M phase. The first stage in the interphase is known as G1. During G1, the cell grows until it reaches its maximum size and also produces many different organelles and many different proteins needed for DNA replication, which moves us on to the S phase or synthesis. During the S phase, the cell uses all of the different proteins created in the G1 stage to replicate all of its DNA. This is done by splitting each of the chromosomes into X-like shapes with two heads called sister chromatids. This way we end up with twice the amount of DNA while keeping the same chromosome count. Now that it's all done with DNA replication, the cell can finally move into the G2 phase. The G2 phase is the final checkup phase in the interphase. During this stage, the cell checks all of its DNA and all of its organelles to make sure there are no damaged parts. If there isn't any damaged parts, the cell goes on to create the more proteins that are needed to enter the M phase. If it is discovered that the DNA is damaged, we move on to something called G0, or the no division phase, in which the cell continues going on its life without preparing for mitosis. The only other case in which we can enter the G0 phase is if there is already enough cells in the tissue and then the cells send a message to the cell telling it not to split so it stays at its size. With all that complete, the cell now finally moves on to mitosis. Mitosis is divided into 5 different stages, starting with the prophase, moving on to the metaphase, then anaphase, then telophase, then finally cytokinesis. It's now time to talk about the prophase. With the prophase beginning, mitosis has officially begun and the division will commence. 3 main things will happen during this phase. First of all, the nuclear membrane surrounding all the chromosomes breaks down and disappears. Second of all the chromosomes condense and coil tightly into smaller, more thick chromosomes. Third of all, string-like things known as spindle fibers start forming at opposing poles of the cell around the centrole. After being done with all that, the cell now enters its metaphase. In its metaphase, the spindle fibers attach from both ends at each chromosome center, known as centromeres, and line them up at the equator or the center of the cell. With the cells all lined up, the cell now moves into its anaphase, or as I like to call it, the splitting phase, during which the spindle fibers shorten, splitting apart the sister chromatids into each side of the pole, so we end up with 46 chromosomes, or whatever the number is in the specific species, on either side of the cell. With an equal amount of chromosomes on either side, the cell now enters its telophase. The first thing that happens in the telophase is the cytoplasm is elongated and starts to look like 2 circles which are only connected by a single side. On each side of the cytoplasm, the cell starts acting as 2 separate cells, in which 2 different membranes form, and the chromosomes in each of these 2 membranes uncoil, the spindle fibers disappear, and the cells are ready to function as 2 daughter identical cells. With the cell already functioning as 2 separate cells, now all that is left is the final round, also known as cytokinesis, which is basically just the cytoplasm splitting down at the middle, causing there to be 2 daughter cells which are identical, and the cycle starts all over again. So that's just about it. I hope you enjoyed the video, and I hope I was able to cover everything needed for this assignment. Have a great day.